Translation. Decoding the message from mRNA to the protein synthesis or the synthesis of a polypeptide in a living cell. This is the first step in a synthesis of a protein. This process is common in both prokaryotes and eukaryotes. What are the steps are involved in a translation? Initiation, elongation and termination. Before initiation, activation of amino acid, charging of tRNA, that is aminoacylation of tRNA. What is aminoacylation? Addition of amino acid to tRNA, that is will be charging of tRNA. That process will take place before these three steps occurs. First one is will be activation of amino acid and then a charging of tRNA. That is, amino acid get activated when combined with an enzyme and they form aminoacyl adenylate enzyme complex. For this it requires energy. Energy can made available by ATP in the presence of a magnesium bivalent ion. It forms aminoacyl adenylate enzyme complex. Now this aminoacyl adenylate enzyme complex join with the tRNA, where this amino acid link with the tRNA, leaving that AMP and a enzyme. Okay, this is said to be charging of a tRNA. Linking of amino acid with the tRNA is said to be charging of tRNA. The first step called initiation starts. Here it requires the three factors, that is IF1, IF2, IF3 in the case of a prokaryotes. What are this IF1, IF2, IF3? They are the protein. Here this out of this three, this IF1 and IF3, this two IF1 and IF3 requires that binding of 30S. 30S is said to be a ribosome. When you take a prokaryotes, the kind of ribosome is a 70S ribosome is present. In this 70S, there are two units, the larger unit called a 50S and the smaller unit called the 30S. This smaller units bind with that mRNA. mRNA is said to be a codon, that is coding strand. At the P site, towards closer to that 3' direction. Okay, this mRNA has got the nucleotide is said to be AUG, that is said to be start codon, where that 30S get a binds here. So here this is said to be a codon and the tRNA should have a anticodon. What's an anticodon for AUG? A can bind with the uracil and uracil can bind with an adenine and guanine can bind with the cytosine. So here the tRNA should have a UAC. This one is said to be anticodon. Okay. As you know that AUG can bind for the methionine amino acid. So the tRNA with the N-formyl methionine IF2, okay, with a GTP that binds at a mRNA and a 30S subunit. Okay. This one is said to be 30S initiation complex. Initiation complex instance where that which initiate that process of translation, where the mRNA codon is present, the tRNA produce an anticodon, IF1 and IF3 that help that binding of a 30S ribosome to that mRNA. Okay. Next this IF2, the another one is said to be IF2, okay, initiation factor 2 that can stimulate that association of a larger unit called the 30S subunit, thereby the larger unit and the smaller unit that form that associate to form a ribosome, that is a 70S initiation complex is formed now. Next step is said to be elongation. Again this elongation is stimulated by three factors EF, TU, TS and G. TU stands for a thermo unstable, this one catalyze and this one help in translocation. Translocation cells that moves that this ribosome started moving on what mRNA stand, that is what a translocation. So all this help in elongation of this process. And then it requires an enzyme called peptidyl transferase enzyme. Now when you take a ribosome, the ribosome are produce the three binding sites for a tRNA. E site, P site and A site. A site is said to be aminoacyl site and P site is said to be peptidyl site and E site is said to be exit site. The first tRNA when they bind with that AUG, that codon, that can enter here at the P site. The second codon onwards they can enter through that A site, aminoacyl site. Now the peptide bond is formed here. The peptidyl transferase enzyme help in formation of a peptide bond between a A side and then a T side. The tRNA present in a A side and tRNA present in a P side. That help in a formation of a peptide bond between a amino acid. Now the deacylated tRNA. Deacylated in sense the tRNA without a amino acid that start moving from a P side to E side, that exit side, and from there it released from the ribosome to the cytoplasm. Okay, the next step is said to be termination. Here, as I said, no, this ribosome started moving towards the mRNA codon stand. Okay, till the A site, no, that reaches that nonsense codon. Nonsense codon are said to be stop codon or the termination codon. What are they? UAA, UAG and UGA. Till that A site reaches a nonsense codon, that elongation process takes place. How they can recognize that termination site? No, it can be recognized by that two releasing factor RF1 and RF2. Once that A site reaches that UAA, UAG and UGA, that nonsense codon which do not code for any amino acid, that is there is no specific tRNA for that. When it reaches that, what happen? No, that polypeptides, no, that get released here, and the tRNA is released from that exit side, and then here what happens here, that ribosome get dissociate, and then mRNA gets separated, and the tRNA which is released, no, they are released in the cytoplasm, and they are ready to pick up the another amino acid and start the process of translation. Okay, once again we'll see what are the steps are involved here. Now the translation. In the process of translation, what happens here? This is the first step in the synthesis of a protein, where that mRNA, the codon present in the mRNA get decoded by a DNA, and then that carry the information to the protein, and which involve the synthesis of a polypeptide chain, and the protein synthesis takes place here in the cytoplasm. Okay. Here there are three main steps are involved: initiation, elongation and termination. Before initiation, what should be done? Activation of amino acid and the charging or the aminoacylation of tRNA. What is aminoacylation? Aminoacylation in sense that the tRNAs get linked with the amino acid. That is said to be aminoacylation. Initiation. If you take initiation, the first step, there are three factors are present in prokaryotes: IF1, IF2 and IF3. And here 9 factors are in eukaryotes. Out of this 3, no, IF3 and here the 9 factors IF2, E stands for eukaryotes. This help in binding with the smallest subunit of ribosome. And here in mRNA has got a codon, and then AUG said be start codon. Here this the ribosomes get bind to that mRNA at the P site of the ribosome, and towards closer to that 3' and AUG is said to be a start codon here, which lie at the P site. Now if you take prokaryotes, the prokaryotes produce the 70S ribosome and eukaryotes produce the 80S ribosome. As you know, the ribosomes that measures by the 20 nanometers, so you have to measure minus 20 here, and it's got a 50 years a larger unit. Again 50 minus 20 is a 30 years is a smaller subunit. Similarly, in the case of 80 years, 80 minus 20 is a 60 years larger unit, and the 60 minus 20 is a 40 years a smaller unit. Okay, now the 30 years that binds with that mRNA with the help of IF3, that is IF3 of prokaryotes, and the 30 years get bind with that mRNA. In the case of eukaryotes, the 40 years is a smaller unit, binds with the mRNA with the help of EIF2. E stands for eukaryotes, and they form the complex, initiation complex here. The anticodon, as I said, mRNA produces the AUG is a codon. The anticodon, the tRNA should carry that anticodon, that is UAC, respective to that AUG nucleotide. And they form a temporary hydrogen bond with the initiation codon, that is AUG represented in the mRNA. Yes, of course, they requires that magnesium bivalent ion for that association. Okay, association of a smaller unit and the larger unit. Now, if you take this, this larger unit, the ribosomes can enclose as mRNA, tRNA complex, that is that occur at the P site. This one is a E site, this one is a P site, and this one is a A site. A site remain exposed here. The ribosome has got a 3 binding sites for that tRNA. What are that? A site is for that acceptor site, okay, where that aminoacyl tRNA can bind. This is for the case of the second tRNA, not for the first one. For the first tRNA to bind with that P site, okay. Acceptor site, that is aminoacyl tRNA binds. What is aminoacyl tRNA? That is a charged tRNA binds there. P site said be peptidyl site, where the tRNA linked, the growing peptide chains occur. E site is said to be exit site, where the deacylated in sense the tRNA without the amino acid get released from that ribosome. Okay. Elongation factors, when you take elongation factors, yes, for the binding, for the initiation factors as well as elongation factors that requires energy gets supplied in the form of a GTP. Okay, and they require the peptidyl transferase enzyme. Peptidyl transferase enzyme help in a formation of a peptide bond between the tRNA present in the P site and the tRNA present in the A site. Okay, that peptide bond is formed here. Next one, translocation. What is translocation? As that yes, when this the ribosomes they started moving towards that mRNA codon till it reaches that nonsense codon. Okay, that is to be translocation. For that requires a translocase enzyme and the energy which is supplied in the form of a GTP. Okay, now what is that tRNA? Deacylated tRNA. Deacylated in sense the tRNA without the amino acid that moves from a P site to, this one is a P site, from P site to E site. Okay, what is it? A site to P site. Here the dipeptidyl tRNA that moves from A site to P site and the formation of a peptide bond between an amino acid. Next step is said to be amino termination. 3 codons. When you take note the 3 codons, this started moving here. This ribosome started moving the A side of the ribosome till it reaches that 3 stop codon or the termination codon UAG, UGA and UAA. With the help of a releasing factor, they can recognize this codon and the dissociation of ribosomes and tRNA and peptide chains. All this take place. That tells about the termination process. Okay. Now we will see what is a site. The review of this the entire process. Where's a site? The site of the translation. Where the translation occurs. The translation occurs in a cytoplasm, where the ribosomes are located. In the case of a prokaryotes is in a 70 years and the case of a eukaryotes is in a 80 years, which surrounds at a mRNA. And the next what is required now is a template mRNA. mRNA acts as a codon stand. Okay, and that codes for the specific sequence of amino acids, which is required for the synthesis of a protein. Translation is said to be the first step in a protein synthesis. Next, what are the requirements for the translation? mRNA, rRNA, that is ribosomal RNA, ribosomes, 20 kinds of amino acids and their specific tRNA. Okay, what are the factors are required here? Initiation factor for the initiation step, elongation factor 1 elongation factor, termination factor, that is for releasing factor. Okay, enzymes. What are the enzymes are required for the translation? Aminoacyl synthetase. This aminoacyl synthetase help in an activation of amino acid before the translation process starts. Peptidyl transferase enzyme help in a elongation process. Peptidyl transferase enzyme help in a binding of peptide bond between a site and then a P site. Okay. And the codons, what are the codons required? Start codon and a stop codon. Start codon is said to be AUG and a stop codon as you seen is UAG, UAA and UGA, which do not code for any amino acid and there are no tRNA for that. That's why it said to be stop codon. Next comes a starting amino acid. In the prokaryotes, what's the starting amino acid? N-formyl methionine. Okay, so by this what we are completing translation process. For the single ribosomes to translate to carry out the translate mechanism, 60 nucleotides, they can produce 60 nucleotides per second. mRNA can read in this direction. There's a 5' to 3'. 5' is said to be upstream and 3' is said to be downstream. It is like a tail region. The polypeptide chain synthesis can occur from a nitrogen to carbon terminal. Okay, what are the steps are involved here in a translation? Once again we'll see that. Review of that. Activation of amino acid, charging of tRNA, and then initiation, elongation and termination process. In a prokaryotes, as you know that it's produced at 70S ribosome, which has got a larger unit called 50S and a smaller unit called the 30S. In a eukaryotes, 80S ribosomes, 60S and 40S. Okay, that's it. That's all about the translation process.